How far can we push the boundaries of biology? This question has sparked the emergence of biotechnology, an exciting field that merges biology with technology. Our story begins in the ancient times, where the roots of biotechnology were unknowingly planted. Yes, you heard it right, unknowingly. Our ancestors, without being cognizant of the science behind it, were already using biotechnology in their everyday lives. They harnessed the power of yeast for brewing beer and fermenting wine, and employed bacteria to make yogurt and cheese. What they were doing was manipulating living organisms to create a desired product, the essence of biotechnology. Fast forward to the 19th century, when Louis Pasteur confirmed that fermentation was caused by living organisms. His work laid the groundwork for the germ theory of disease, catapulting us into a new era of scientific understanding. The mid-20th century brought about another monumental discovery. In 1953, James Watson and Francis Crick unraveled the double helix structure of DNA, the blueprint of life. This revelation was the turning point that would forever change our understanding of heredity and life itself. Following this, the 1970s marked the birth of genetic engineering. Scientists Herbert Boyer and Stanley Cohen made the first ripples in this new pond. They successfully spliced a gene from one bacterium and inserted it into another, creating the first genetically modified organism. This was the dawn of a new era, where we could now modify the very code of life. Genetic engineering opened a Pandora's box of possibilities, laying the foundation for modern biotechnology. It propelled us into a world where we could not only understand, but also manipulate the genetic makeup of living organisms. This has led to breakthroughs in medicine, agriculture, and environmental science, to name a few. From humble beginnings, biotechnology has grown into a revolutionary science, promising to reshape our world. The journey of biotechnology from ancient brewing techniques to genetic engineering showcases humanity's relentless quest for knowledge and progress. But remember, we're just at the dawn of this exciting field. What lies ahead is a future brimming with unimaginable possibilities. The discovery of DNA's structure opened the doors to the world of gene editing. From the moment the double helix of DNA was first visualized, the idea of manipulating our genetic code became less of a science fiction dream and more of a tangible reality. Early gene editing techniques, like recombinant DNA technology, gave us our first glimpse into this potential. Scientists could cut and paste fragments of DNA from one organism to another, creating the first genetically modified organisms. But it was the advent of targeted gene editing techniques that truly revolutionized the field. Techniques like zinc finger nucleuses and TALENs allowed scientists to target specific genes, but these methods were often expensive, time-consuming, and lacked precision. Then, the landscape of gene editing was forever changed with the discovery of the CRISPR-Cas9 system. Derived from a naturally occurring defense mechanism in bacteria, CRISPR-Cas9 brought a level of simplicity and precision to gene editing that was previously unimaginable. With CRISPR, scientists could now program the Cas9 enzyme to cut the DNA at a specific location, allowing them to add, delete, or replace genes with ease. This revolutionary tool has allowed us to edit the genes of organisms with unprecedented precision, from plants and animals to even human cells. The implications of this technology are vast. We've seen the potential to cure genetic diseases, improve crop yields, and even modify organisms to better adapt to changing environments. We've begun to unlock the secrets of our genetic code, and with it, the potential to shape our own evolutionary path. But as with any powerful tool, gene editing comes with its own set of ethical questions. How far should we go in editing our own genes? Who gets to decide what changes are made? And what happens if these changes have unforeseen consequences? Gene editing has become a powerful tool in the arsenal of biotechnology, offering the potential to rewrite the code of life. As we continue to grapple with these questions, one thing is clear. We stand on the brink of a new era in biotechnology, one that promises to reshape our world in ways we can only begin to imagine. Beyond gene editing, biotechnology has ventured into the realm of synthetic biology. This is a field that goes a step further than merely understanding and manipulating genetic code. It's where biology meets engineering, where life becomes a canvas for design and artistry. Synthetic biology is a fascinating fusion of disciplines. It involves designing and constructing new biological parts, devices, and systems. Think of it as an exciting new frontier where we are not just observers of life's intricate machinery, but active participants shaping and guiding its course. One of the most intriguing applications of synthetic biology lies in the production of biofuels. Scientists are reprogramming microorganisms to convert plant material into fuels. It's like having tiny factories that take in green waste and churn out eco-friendly energy. Imagine the possibilities when we scale this up, but it doesn't stop there. Synthetic biology is also being harnessed for environmental cleanup. Take the example of bacteria engineered to consume oil spills. These microscopic janitors are designed to devour pollution, potentially offering a sustainable solution to some of our most challenging environmental problems. In the realm of medicine, synthetic biology is a game changer. It's being used to develop new treatments for diseases, even those that have long eluded us. From designing bacteria that can hunt down and destroy cancer cells to creating synthetic vaccines, the medical potential of this field is truly staggering. But of course, with great power comes great responsibility. The ethical implications of synthetic biology cannot be understated. As we step into this new era, we must tread carefully, balancing our desire to innovate with the need to respect and preserve the delicate balance of life. Synthetic biology is paving the way for a new era of biotechnology, one where we can design and engineer life to our specifications. As we continue to push the boundaries of what's possible, who knows what breathtaking innovations lie just around the corner? Stay tuned as we delve deeper into the future of biotechnology. As we look to the future, the possibilities of biotechnology seem limitless. Imagine a world where personalized medicine is not just a concept, but a common reality. Picture a time when doctors can treat patients with therapies tailor-made for their unique genetic makeup. This could mean more effective treatments with fewer side effects. It could mean the end of a one-size-fits-all approach to medicine. Biotechnology holds the potential to make this vision a reality, and it's closer than we may think. Now let's push the boundaries even further. What about designer organisms? Synthetic biology could allow us to engineer organisms, from bacteria to plants, to perform specific tasks. These tasks could be anything from cleaning up oil spills to producing biofuels. The possibilities are as diverse as life itself. But perhaps the most tantalizing prospect of all is the potential to extend human lifespan. Research is already underway to understand the genetic factors that contribute to aging. With the power of gene editing, we may one day be able to slow down or even reverse the aging process. Imagine living a longer, healthier life, free of age-related diseases like Alzheimer's and heart disease. Yet, with these incredible possibilities come significant ethical challenges. How will we ensure equitable access to these technologies? How will we prevent misuse? And perhaps most importantly, should we even attempt to alter the course of natural life and evolution? As we stand on the brink of this new frontier, it's essential to engage in robust ethical discussions. We need to consider not just what we can do, but what we should do. We need to balance the potential benefits with the potential risks and make informed decisions about how to proceed. The future of biotechnology is a vast frontier, filled with potential and fraught with ethical challenges. The journey ahead is exciting, filled with promise and potential, but the path will not be without its hurdles. As we stride forward into this brave new world, let's do so with our eyes wide open, prepared to face the challenges that lie ahead. From its inception to its promising future, biotechnology has come a long way. As we trace back the journey of biotechnology, we find roots entwined with ancient practices. Long before the term biotechnology was coined, our ancestors were experimenting with it, using yeast to ferment beverages, and selectively breeding plants and animals to accentuate desirable traits. This was biotechnology in its crudest form, laying the foundation for the sophisticated techniques we wield today. As time progressed, so did our understanding and manipulation of biological processes. The 20th century witnessed remarkable strides in biotechnology. The discovery of DNA structure, the advent of genetic engineering, and the Human Genome Project are monumental milestones that have shaped the trajectory of this field. Fast forward to the 21st century, we're living in the era of gene editing and synthetic biology. Revolutionary tools like CRISPR-Cas9 have demystified the complex language of life, allowing us to edit genes with precision never thought possible. Synthetic biology, on the other hand, is enabling us to design and construct new biological parts, devices, and systems, pushing the boundaries of what is naturally possible. These advancements are not just confined to theory and research, they're beginning to permeate our everyday lives, promising transformative potential. From disease eradication to personalized medicine, from sustainable agriculture to biofuels, the possibilities are as vast as they are exciting. Yet this journey is far from over. We're standing on the threshold of a new era, an era brimming with potential and challenges. As we delve deeper into the DNA's secrets and engineer life forms in ways never imagined, we're reshaping not just biology, but our very understanding of life and existence. As we stand on the brink of a new era in biotechnology, we invite you to join us in exploring this fascinating field and its potential to reshape our world.